Welcome to this explainer. If you want to know exactly how scientists are decoding human genetics to actually fight disease, well, you are in the exact right place. Today, we're taking a really structured, energetic dive into next-generation sequencing, or NGS. Specifically, we're going to navigate the critical differences between somatic and germline variant calling. By the end of this explainer, you'll understand the distinct laboratory workflows we use to detect hereditary disease risks versus the actual drivers of cancer progression. So let's just get right into it. Here is our roadmap for today. We'll start with the shared foundation of NGS, branch off into germline calling, and then tackle the chaotic complexities of somatic calling. We'll explore the VAF sensitivity gap, break down somatic filtering, and finally summarize the protocols side by side so you have the whole picture. This illustrates the complete map we're going to be navigating today. Notice how both workflows start right together at the top under a shared pre-processing phase, but then they diverge sharply down two entirely separate paths, all depending on whether we're hunting for inherited mutations or acquired cancer mutations. We are going to break this entire map down, piece by piece. First up, the shared NGS foundation. Okay, so whether you're searching an entire genome for inherited traits or isolating a specific tissue for cancer mutations, every single sample identically undergoes a foundational pre-processing phase. This shared foundation consists of two main steps: DNA alignment, which is basically like mapping billions of tiny puzzle pieces to a master picture, and quality recalibration, which filters out technical typos. It's only after the shared groundwork is laid that the workflows actually split. All right, let's take that left path into germline calling. Germline calling acts as a sort of baseline control for human genetics. Here, we are searching for inherited mutations. Because these variants are passed directly from an individual's parents, they're fundamentally woven into their biology. This means these mutations are found uniformly in every single cell of the human body. And you know, that uniformity, it is absolutely crucial for how we detect them. Now, before we look at the exact tools we use, we have to define a really important metric: variant allele frequency, or VAF. Think of VAF as a signal strength indicator. It's simply the percentage of sequence reads carrying a specific mutation compared to the total number of reads at that exact location. It basically tells us just how prominent a mutation is within our sample. So how does VAF actually apply to germline variants? Well, because germline variants are inherited and present in every cell, they have a very high confidence, mathematically clean VAF. For a heterozygous trait, meaning you inherit a mutation from just one parent, the VAF typically shows up right at 50%. If the trait is homozygous, inherited from both parents, the VAF hits exactly 100%. It is a clean, easy-to-spot signal. Because these 50% or 100% signals are so prominent, we don't need to hunt for faint whispers in the data. That's exactly why the industry standard primary calling tool for these high-confidence inherited variants is the GATK haplotype caller. It's perfectly optimized to confidently identify these well-represented, expected genetic variants. Okay, let's flip over to somatic calling. Let's jump to the second, far more complex path. Unlike the inherited traits we just looked at, somatic mutations are acquired over a person's lifetime. They are not in every cell. Instead, they're isolated only to specific tissues, like a tumor in the lung or the liver. And what makes this incredibly challenging is that these mutations are hidden deep amidst tumor heterogeneity, meaning a tumor isn't just one uniform mass of mutated cells. It's this chaotic, constantly evolving mixture of mutated cells, normal cells, and subclones. It's basically a biological blender. Which brings us directly to the VAF sensitivity arc. This is the classic massive needle in a haystack problem. Dramatically contrasting that clean 50% germline split, a somatic low-frequency variant often exists at a VAF of less than 5%. Because of that dense tumor heterogeneity, the mutated DNA is heavily diluted by the DNA from surrounding normal cells. You are literally looking for a tiny fraction of a signal within a massive background of healthy tissue. And this matters immensely. If we miss this tiny fraction, we might miss the very mutation driving a patient's cancer, the exact target we need to select the right life-saving therapy. So our standard germline tool, the GATK haplotype caller, it simply won't work here. It would miss those faint signals entirely. To detect a VAF below 5%, we must switch to ultrasensitive specialist tools. In the somatic workflow, we rely on primary calling tools optimized specifically for low-frequency mutations, heavy hitters like Mutec2, Strelka2, and Vardict. These algorithms are engineered exclusively to find that needle in the haystack. But that leads us to a new challenge: somatic filtering. Here's the catch. Deploying ultra-high sensitivity introduces a massive new problem. Because these specialist tools are so incredibly sensitive to faint signals, we're forced to introduce immense pipeline complexity to filter out the noise. Let's look at the 3 hurdles we use to filter out this noise, ensuring we don't accidentally flag a random lab error as a cancer mutation. First up, we use a matched normal comparison. To know for sure that a mutation in a tumor is an acquired somatic mutation driving the cancer, we ideally compare the tumor sample against matched normal tissue from the exact same patient. By comparing the two, we can safely subtract the patient's natural inherited germline background, leaving only the newly acquired tumor mutations behind. Next, we tackle lab noise through artifact removal. You see, during the process of preserving and sequencing tumor samples, DNA can get chemically damaged. Things like the formaldehyde used to preserve the tumor tissue, known as FFPE, can actually chemically damage the DNA, creating fake signals that look exactly like low-frequency mutations. So we deploy specialized machine learning filters designed specifically to recognize and scrub out these false, chemically induced signals. Finally, to maximize our overall precision, we use ensemble methods. Look, even with great tools like Mutec2 or Strelka2, no single caller is perfect. By using a tool like Somatic, which intelligently combines the results of multiple different variant colors, we cross-reference their findings to achieve significantly higher F1 scores. This means our final list of cancer mutations is as reliable as humanly possible. Let's bring it all together for a protocol comparison. This perfectly captures those diverging paths we've been talking about. Germline calling aims to identify inherited disease risk, while somatic calling seeks to detect the active drivers of cancer progression. Germline operates on a high, mathematically expected VAF of 50% or 100%, whereas somatic workflows require ultra-high sensitivity to find those elusive variants below 5%. And for the control sample, Germline needs just a single sample, but somatic heavily prefers matched normal tissue to confidently filter out that inherited background. As we continue to push the boundaries of personalized medicine, tumor mutations are becoming ever more elusive to track. We have incredible algorithms today, but as we seek to detect cancer earlier and earlier from fainter and fainter signals, we have to ask, what advanced AI-driven tools will drive the next great leap in low-frequency cancer detection? It's a fascinating space to watch. Thank you so much for exploring this with me today, and keep learning.